be a review for most people. The types of synapses, the majority of the synapses in the mammalian system, or the majority in the parts of the brain that we mostly look at, in any case, tend to be chemical synapses. There may be more electrical synapses than we know of, though, even in the cortex. And again, I referenced Roger Traub's notion that the axo-axonal gap junctions are very important. And he hasn't gained a lot of traction with that, but that could be the case. So chemical synapses themselves, of course, come in many varieties. And the main dichotomy there is between ionotropic and metabotropic, where ionotropic let ions through, so that when a neurotransmitter is received, ions enter, either exciting or inhibiting, giving an EPSP or an IPSP, a depolarization or a hyperpolarization, respectively, to the postsynaptic location, whether that be a soma, a dendritic shaft, or a spine. Meanwhile, the other type would be metabotropic receptors, where what would be released internally would not be anything coming from the outside, but would be something synthesized or broken down from the inside, such as cyclic AMP, a G protein, or other. And those then might go on to activate channels. So a metabotropic receptor can also give you an EPSP or an IPSP, or it can be a purely chemical process. Perhaps, most likely, even chemical processes that are not local but reach down as far as the soma and the nucleus to change the expression of genes. So enormously complex. In this chemical synapse, we see only the unidirectional, but there's also evidence of back transmission, typically thought to be via endocannabinoids. The way the action potential causes all this to happen is that the depolarization in the presynaptic specialization, the part on the top, let's see, I've got a pointer there. There's a little picture of an action potential. That depolarization causes entry of calcium, probably other ions, probably sodium as well, but calcium then has to do as a second messenger with the melding of the vesicle with the presynaptic plate. Neurotransmitter proceeds across the synaptic cleft and have the effects we just described. And an electrical synapse, of course, it's simply that some current passes through in one direction or possibly in the other direction. Again, the types of chemical synapses include ionotropic, where we have direct passage of ions through pores in the same way, in a similar way, to what we see with the voltage-sensitive channels, except these are not voltage-sensitive, but are sensitive to a chemical, to a ligand. Versus, on the right, the metabotropic, which can then secondarily give connectivity to an ionotropic, something that will give ion passage. Interestingly, a lot of these effects may be on the constituent voltage-sensitive ion channels of the membrane. And in particular, we've had a lot of interest, a lot of other people as well have had a lot of interest in IH, the HCN, or hyperpolarization activated channel, or it's got a bunch of names because it's anomalous, the anomalous rectifier, the funny current, the queer current, it's all these names, also found in the heart, that are affected by cyclic AMP, and we have a paper on that, or even two, I think. The complexity, of course, grows and grows, and we do not get any of this complexity in our models so far. And in fact, part of our submission for the renewal for our reaction diffusion grant, which is headed up by Mia and by Robert, is to really start looking in more detail at these synapses, and we're working with a guy at USC to do that. But as you can imagine from what you've already seen, doing this at any level of detail is going to be tremendously computationally expensive. And in fact, M-Cell does this in tremendous detail. That's Tom Bartol's program, which follows individual ions, individual molecules, and he has these these enormously HPC-intensive program to do that. But some of the things that might be considered are these various steps: the synthesis of transmitter, its storage in vesicles, the number of vesicles that are stored, the number of molecules within a vesicle, the likelihood of melding of the vesicle with the presynaptic membrane in the context of particular degrees of depolarization. Because remember, even though the action potential is all or none, there is evidence that altering the membrane potential of the entire cell from the soma can actually alter the release of transmitter. Then we could consider the types of calcium channels that are present in the presynaptic space, the way that calcium enters, how that calcium has its effect on the vesicles. We could consider the effect of going across the synaptic cleft. And in fact, we've begun to consider that in a very simplified way by thinking about what are the effects on cleft transmitter receptors, cleft receptors versus extrasynaptic receptors, which would be receptors that are lying on the sides over here. And there's always or often evidence of spillover, particularly of glutamate, with very strong stimulation, which can give you activation of extrasynaptic as well as intrasynaptic receptors. Then postsynaptically, again, metabotropics have tremendous complexity of cascades, but even ionotropics, we can think about the activation, the time constants of that activation, the flux of calcium and sodium, for example, through an NMDA, and the effect of that at both local receptors that are also present nearby postsynaptically, and if in the case of a spine, also how much of that might get through the neck and affect the rest of the cell. Here, yet another picture showing much the same thing. In this case, presumably an Ampa receptor, the major glutamatergic excitatory receptors in the cortex are thought to be Ampa, which is fast, and NMDA, which is somewhat slower and longer acting, and also has the potential, has linkage with the degree of depolarization postsynaptically. So this possibility of what would be considered a Hebbian association, that postsynaptic depolarization and presynaptic activation by transmitter would be required to get flow of, in that case, sodium and calcium through the channel. So the action potential presynaptically, release of, I don't know why it's release of calcium. It should be release of transmitter. I'm not sure what that calcium is doing there. I think that should be intracellular. And then the release of transmitter, and then the EPSP postsynaptically. So EPSP, excitatory postsynaptic potential, depolarizing, going to a potential high, less negative than the resting membrane potential of about -65, and inhibitory, a hyperpolarizing membrane potential taking you to a potential more hyperpolarized, so something like -70, -80 potential. And there are also what are called shunting potentials, which in the absence of any other signal you wouldn't even see, because all they are is opening up channels that are changing the input resistance of the cell without changing the polarity of the cell, without depolarizing or hyperpolarizing. And so those shunting effects, which you can see mostly with GABA-A chloride synapses, would now cause any signal, whether it be excitatory or inhibitory, to have less effect because it will simply leak out of those channels. So it's called shunting potentials. So here is just pretty pictures and excitatory postsynaptic potential. It's as if you could activate the presynaptic specialization on its own with an electrical stimulator. You can in the squid giant synapse, but this is not a picture of squid giant synapse, so this is imaginary. And then inhibitory, much the same, except now you have a negative going potential. You can now, however, do something just like this because we have channel rhodopsin, which is exciting. And this is a figure from Salvador's work where we looked at data from Ben Suter and Gordon Shepard Jr. to identify how we could map the circuit with what's called SCRACM, which is subcellular channel rhodopsin associated circuit mapping. And so here we have a particular input into this type of cell, which are the pyramidal type cells of layer 5 of mouse primary motor cortex. And we ask what is the density of activation at different locations on this cell. And we can see that these basodendrites that are kind of almost apical obliques because they're pointing up towards the apical, these are definitely apical obliques, and some of this apical dendrite have this activation. The way this experiment is done is this channel rhodopsin is initially injected into VL thalamus, ventrolateral thalamus, and then passes anterograde to the presynaptic specializations. So now you have presynaptic specializations that are sensitive to the laser light, which will then cause a depolarization in the presynaptic specialization, and you can measure with an electrode in the soma of the PT cell where the activation is producing a EPSP in this case. So they scan a laser across these various sites and say we have a lot of activation here where it's bright red and a moderate amount here where it's green, and really here in the tuft, no activation. Here in some of the basodendrites, no activation, and others, a little bit of activation. And we can then compare, well, how is this projection onto the cell from ventrolateral thalamus compare the projection on the same type of cell, not the same individual cell, because it has to be a separate experiment with injection of channel rhodopsin in S2 now instead of VL thalamus. So it may just be differences between individual cells that we're seeing until we get a very large N. And that's difficult to get, as you can imagine. Reality is always far more difficult than simulation, which is why I don't do it. So that you can activate here and see that there's perhaps overall somewhat lesser activation and a somewhat different pattern here. There's this more distal activation from S2, a little bit more proximal from ventrolateral thalamus in the apical dendrite of the pyramidal type cell. Any questions there? So I think it's very cool, and it's very much your future, if you stay in this field, is to really get an idea of where projections go on cells. A key point is that spatial summation is typically nonlinear. It's often highly nonlinear, and this really is, in these figures, they're assuming passive membrane, but we talked earlier, there is no passive membrane. And so once you have active membrane, the nonlinearities, if anything, are exaggerated from what we'll see here. So the idea is if you stimulate location 1, stimulate location 2, you get 2 EPSPs of comparable size. And then if you stimulate both together, you get an EPSP, in this case, that is not the sum of the two. And that's a variety of factors in the passive condition, mostly having to do with leak factors. So the spatial, the nonlinear spatial summation has to do with the reversal potential for that ion. We talked about sodium being plus 55, potassium being minus 85 or so, to get a driving force. And when we talk about the shunting potential, the driving force is the same, but the conductance, the local conductances are much different. So the driving force will be lower as the membrane voltage of it. The driving force for an EPSP will be lower in the conditions of a prior EPSP. And it also depends on the timing. At one EPSP, the first EPSP will tend to reduce the driving force of the second. And there's also the shunting effect. We talked earlier about backpropagation and the idea that backpropagation can give you those complex patterns of activation. Here's something on shunting inhibition with inhibition misspelled, sorry. EPSP and IPSB IPSB cancels the EPSB effectively in the case of shunting inhibition. The shunt is considered an IPSB because it reduces other IPSBs. It also reduces EPSBs by simply allowing the current to leak out of the cell. You can understand that with a pyramidal cell, particularly those enormous ones from layer 5, which in the human can be the Betz cells, which are really practically visible to the naked eye, excuse me, if stained correctly, that you can get all kinds of patterns of temporal and spatial summation, given that you probably have order 10,000 excitatory synapses onto these cells of various types, AMPA NMDA, mixed AMPA and NMDA, GABA A, GABA B, peptides galore, and just enormous complexity, as well as norepinephrine, dopamine, serotonin, others. So just an example here of summation versus no summation, with the potential for activation of a dendritic or a somatic action potential, and a potential for IPSP canceling an EPSP. This is something that we've looked at a fair amount, working with an experimentalist, Surgeon Antic, and this is work that was done by Penny Gao and by Joe Graham, who's teaching this class. We looked at how backpropagating action potentials would be affected by NMDA plateaus. NMDA plateaus can last 100 to 400 milliseconds. You can activate them in the dendrites, and they give you this platform on which you can get more dramatic spiking. In this case, the EPSP by itself, giving just one EPSP, two EPSPs. The plateau by itself giving this plateau. That's about, what, 150, maybe 200 milliseconds. And I don't have the voltage bar, but I'd say it's about 40 millivolts depolarization. Oh, there it is. So 20 millivolts depolarization. Now added together, EPSP and then the EPSP during the plateau. This is without the EPSP during the plateau. Is it? What's different between here and here? Sorry, plateau only. Okay, this is just low, right. This is gives you a spike. And now with a greater activation, you'll get plateau activated spikes, but more spikes with the EPSP plus the plateau. So get these complex combinations. This is an idea that we brought forth, and that is independently brought forth by Hawkins in his new book. He's that guy who founded some major company, I forget which, and now writes books about neuroscience, has this Neuroscience Institute. Anyway, the notion is that these plateaus in the dendrites could give you prepared cells, where the prepared cell is reflecting a Bayesian prior of what you might be expecting to sense or identify cognitively even. And having that Bayesian prior then makes EPSPs more likely to fire the cell. So this is a popular and cool idea with respect to functionality. Briefly, how are synapses handled in neuron and in NetPyNE through neuron? The presynaptic cell simply has to announce a spike. It spikes, and it adds that event, the spiking event, to the queue. And there's a main queue, and that is going to be delivered postsynaptically after a delay. And the queue can be used for a variety of things. We would often use it, for example, when we might want to do several steps of eye clamp to start and stop eye clamps. We can use it in some of these artificial cells to start and stop spiking, as is done with NetStim, for example. So there's a lot of uses for the queue. And often when you get to debugging a complicated situation, you may want to print out the queue, which is all accessible to you from neuron. So the synaptic connection model actually in neuron includes three parts, and Salvador is going to go into this more as we go through the tutorial a little later. The presynaptic cell part is not something that you typically have to identify or access on its own. It's a monitor that is looking for voltage, typically voltage, although you can measure calcium, you can measure anything as your presynaptic indicator, looking for something to go past a threshold to give that signal that will go onto the cue. The way you set that threshold is not directly, but is in the NetCon, network connector. And the NetCon then is the line, and that has a weight, which is nominally the amount of neurotransmitter. It's the strength of the synapse, delay, the time for the spike to reach. Often in NetPyNE, we'll set the delays depending on the distance between the cells. And I didn't put here, but it has that threshold, which tells you what number to look for. If you don't put the threshold in, it has a default, I think it's negative 10 millivolts, so it will generally pick up spikes and work the way you might want it to as a default value. So the one thing you have to set up is the NetCon. The other thing you have to set up is the synapse itself. The synapse lives on the postsynaptic cell, of course. It can be on the dendrite, it can be on the stoma, wherever you want to put it. It would be something like an XPsyn, EXPSYN, that's a mod file, or X2syn, or Ampa, or an NMDA. These are all mod files. And so you insert them. No, I'm sorry, you don't insert them. Insert is for suffixes. For the point process, you just simply create them. So you say something like newsyn equals H dot exp to SYN, open paren, dend 0.5, dend 0.5, close paren. So to tell where it's going to go. This will typically have an equilibrium potential. So if you set the equilibrium potential to positive 10 millivolts, that's going to be an EPSB. If you set it to negative 90 millivolts, that's going to be an IPSB. For X2syn, it'll have a time constant. The parameters will be a little different for AMPA and NMDA. The NMDA will also include a magnesium level for magnesium blockade. The X2syn will include a tau1 for up and a tau2 for the decay time. It changes a conductance G based on the weight that comes from the NetCon. So that's a little confusing sometimes because the weight is coming from the NetCon. It's not intrinsic to the synapse in the way that it would be for a voltage-sensitive channel. And here we have the EPSP versus IPSP. So that's the sort of pattern of activation, the way we set up. And instead of a presynaptic cell, one can set this up as a NetStim, which simply sends a stimulation event. It does not simulate voltage. It does not have to look for a threshold. It simply sends the event at a certain time that you choose. It can send multiple events. You set the number of events. You set the noisiness of events. It's worth mentioning that the way XPsyn and X2syn are set up, as well as some of the AMPA and NMDA and GABA-A synaptic mechanisms, you can have 2, 3, or 100 or 1,000 NetCons activating the same location, the same dendrite. And that's actually saves a lot of simulation time to not have these redundant, well, it saves space. I think it also saves a little simulation time, not have these redundant synapses. I discussed this about NetPyNE. I'm not sure if this ever got incorporated in NetPyNE. It probably doesn't really make a great deal of difference for most simulations, but it might in some cases. But just a generic network to just give some ways of thinking about networks. So this is kind of as simple as it gets. We have here, we can call our neurons, our units. Here I called them 1, 2, 3. I think I'm about to change to A, B, C, D. So somebody's slacking me, but saying that they hope I'm ignoring Slack. Okay, now I'm ignoring Slack. So A, B, C, and then we have connections. And there is something wrong with this picture. I don't know if I can find it. Maybe there's not something wrong. Ideally, we generally, here it is, expect to follow what's called Gale's law, that a cell either is GABA in the cortex, not in other areas of the brain or in other areas of the body, is expected to be either excitatory or inhibitory, but not both. So you expect everything coming out of a cell. Here, cell 8, everything coming out should be positive or negative. So this is a little anomalous in terms of our understanding of cortical circuitry, although it could be the case that other circuits would allow this kind of positive projection here. 0.7 is the weight versus negative projection, negative 0.4. So out of that, what we'll often work with is some kind of a connection table. Often it's a sparse matrix. One can set this up in MATLAB, Python. As they get bigger, of course, you don't draw them, and if you look at them, you look at them with a color coding rather than these numbers, because if you've got a 10,000 cells and you have a 10,000 by 10,000 matrix, and you have 10,000 of some other type of cells, so you have another matrix, and then you have another matrix. It can be quite overwhelming. But here, in the simple case we gave, we have the presynaptic here, as I mentioned, listed as ABCD cells here, and the postsynaptic as ABCD here. And listing it in this way is convenient and nice and works through with linear algebra, because it's the same as the connection matrix that you would multiply by presynaptic states in a very simple artificial neural network to get a set of postsynaptic states. So that's why this particular layout is nice, the from across columns and the to across rows. And then we can just talk very briefly about the algebra of synaptic connectivity, just to see it once again, see it twice, see it 5 times. You'll be familiar with it. We can call S the number of synapses. The divergence, D, means how many outputs come from a given cell. Again, this may be thousands in a cortical model. Convergence, how many inputs come into the cell. In the case of cortical models with realistic dendrites, we also have to worry about convergence at different locations on the dendrite versus location on the soma versus basilar dendrites versus soma apical dendrites. So we have, again, enormous complexity. We can calculate the total number of synapses, let's say converging onto the soma by the convergence onto the soma times the number of postsynaptic cells projecting to a cell or a set of cells. Similarly, the number of synapses will be the divergence times the number of presynaptic cells. And we often see reported in the literature, or we'll report ourselves, a percent, a probability of connection, which is the total number of synapses via the total number of synapses over the total number of possible synapses, which would assume that every pre is attached to every post. So it would be number of presynaptic cells times the number of postsynaptic cells. Algebraically, this works out from C over pre to D over post. And it's slightly different if you're talking about how a population acts on itself, assuming you don't allow what are called autopses, A-U-T-A-P-S-E, autops, which are synapses on yourself. Those do occur certainly in culture and probably occur in real life as well. So it's not, maybe it's not a bug, but it's probably not a feature, but maybe it is, who knows? So
Sorry,
cortex.
Bill?
Yeah.
I just got a little bit lost, but is there any paper that shows this exactly, algebra or anywhere where I can find more information about this?
I think this might be in one of my early papers.
Okay.
But I mean, you will have access to these slides if you just want to look at this slide more closely and work it through.
Okay, thank you.
No problem. So Cortex has been, remains the most popular site for modeling, and that's because we humans are excessively proud of our cortex. Now, I try to be proud of my thalamus. I try to be proud of my basal ganglia. I have all kinds of wonderful deep telencephalic parts that just are underappreciated. But the cortex is everybody's favorite. What can I tell you? And it's ironic that people say, oh, we're modeling the brain, when really what they're modeling and what they're studying in many experimental cases is just cortex. The brain, obviously, most of its volume is not cortex. Anyway, be that as it may, it's very corticocentric, anthropocentric, corticocentric. So we have these layers of cortex, which are thickness, which may be approximately 1 millimeter in a mouse, maybe 5 millimeters. Salvador will correct me if I'm wrong, in a human or a macaque. We have sulci and gyri. It's folded. It's more folded in us than it is in many of the other primates. In particular, some of the primates are practically what's called lissencephalic, smooth cortex, not folded. That's actually an anomaly in some human babies that are born with severe brain deficiencies, lissencephalic brains. The ventricles are in the middle, as pointed out here. The cortical layers are a notion that came from a guy, Brodmann, again, a century ago. This stuff just persists and persists and persists because we don't get better ideas. And he came up with this idea that there's 6 layers. Well, are there 6 layers? Maybe some places. Sometimes they can be hard to identify. Sometimes you end up, as in this visual cortex, saying we're going to call it 4A, 4B, and 4C. So we got suddenly 2 more layers. When we go to motor cortex, we almost always subdivide 5A and 5B, which then start to be very broad. This would be layer 5 would be very expanded in motor cortex, and layer 2 and 3 would be so small that they'd be considered layer 2/3. So they kind of go together. Layer 1 is very interesting. It's not acellular, but it's many fewer cells. The cells that are there are mostly inhibitory. Long projections and some of the thalamic projections from what's called the matrix of the thalamus project into layer 1. They reach some of the tufts of these layer 5 cells. This is a total mystery. You have thick tufted cells, you have thin tufted cells. It's not clear how those signals get through, how they are used, how they summate. It's cool. I'm sure it's cool. I just don't know what it is. That's all I know is it's cool. Cortex differs across mammals, or if you happen to be trying to sell your grant to NIH and you're doing a rat or a mouse, you say, oh, they're very similar across mammals. So it's all depends on your perspective. Of course, a lot of research is done on mice, and we try to make the comparison to humans, and we do the knockouts to emulate human diseases such as Alzheimer's disease, autism, Huntington's disease, Parkinson's disease, and we say, well, the mouse is similar enough. Mouse has become very popular lately because it's so easy to crossbreed them. We get mice from Jackson Laboratories with all kinds of particular genes in them, and there's a lot of tools available to identify transcriptomics, etc. The rat less so, and I guess the rat's more variable in terms of different rat species. Where the rat was popular for a long time because it's got a bigger brain than a mouse, so it's easier to work with electrophysiologically. The macaque is another organism that, of course, is popular. I don't even know. Let's see how big the layer 4 is. The layer 4 is not enormous here, so this is probably not primary sensory cortex. I thought I just saw a Slack come through, but it disappeared. Okay, so layer 1 is this mystery layer of projection, long projections, inhibitory cells. Layer 2 here divides 3A and 3B, so a lot of layers here. Now I've got myself curious. Here, so about 3 millimeters here, 1 millimeter here, are pyramidal cells, but not enormous pyramidal cells, as well as many inhibitory cells. Layer 4 would be the layer where sensory cortex would receive inputs from thalamus, core thalamus in this case. There is also a layer 4 in motor cortex. It is discovered by Gordon Shepard Jr. It's not the same shape of cells, but it's the same properties of cells and the same connectivity. Layer 5 is the one that we've investigated the most, have these enormous cells, particularly in motor cortex. Again, in motor cortex, this would be a much larger layer. 5A and 5B shown here in the rat. Layer 6 have projections in particular to thalamus, the corticothalamic cells. So, and then within the cortex there are these well-known projections. So the canonical model. I get in trouble for some reason with Salvador, not with Salvador, not probably with Salvador as well, with Gordon Shepherd Jr. for saying canonical model. This is a Douglas and Martin notion that became very popular. Projection into layer 4, particularly for sensory cortex, projection up from layer 4 to layer 2, 3, projection down from layer 2, 3 to layer 5, being a canonical pattern of connectivity, which does seem to be true, but is a very simplified view. Here is another thing that is a debatable phenomenon. So cortical columns have primarily been identified physiologically. The original column definitions were from Mountcastle and somatosensory cortex, and then there were similar definitions in visual cortex by Hubel and Wiesel. And this would date us back to the late 50s, early 60s. What identifies a cortical column physiologically is that there is a pattern of stimulation that's characteristic of that column. So, for example, in vision, you have ocular columns. You have a column that gets mostly from the right eye. You have a column that gets mostly from the left eye. You have a column that gets mostly particular oriented bars, as defined by rate coding, which is another question. So I just pile up the questions here, don't I? The annoying thing about columns is that they, by and large, cannot be identified anatomically. So how big is a column? Well, it depends on your organism. In fact, if you're looking at a mouse, it's very hard to identify columns, visual columns physiologically. You can't identify them anatomically, so you really don't know. Are they even there at all, or is it just kind of mush, kind of a sliding Gaussian of connectivity across cortex without any discretization? Again, the notion of the column would be it should be discrete in some way across cortex. So it would include all the layers, but going across tangential, it would have a beginning and an end. The one place where you can clearly see anatomical columns is the whisker cortex, where you have these barrels in layer 4, which are very obvious and stand out. And you can see this barrel attaches to this whisker, and that is the one place that columns are really clear. But we use the notion, and we say we're simulating a column because it's a popular notion. But you can read a lot of the anatomical literature about, do columns exist? Are they microcolumns? Are they columns? Are they nanocolumns? How big are they? We don't know. I don't know. Different cells across the cortex are different characteristic shapes. Again, our favorites tend to be these massive L5 cells. The L5A cells tend to be these intratelencephalic cells, which are thin, tufted, and are relatively adapting. The somewhat lower L5B cells are thick, tufted, and more strongly firing. These are the so-called pyramidal type cells, or corticospinal cells that project out of telencephalon. They project out to brainstem. They project out, in the case of motor cortex, even all the way down to spinal cord. But other areas don't project spinal cord, just to brainstem. Intratelencephalic cells, these cells will just project to other parts of telencephalon, often to the other side, the corresponding cortex on the other side of the brain, across the corpus callosum, as well as to striatum. In fact, both project to striatum. One projects ipsilateral striatum, the other projects contralateral striatum. Layer 2, 3 cells are also pyramidal cells. Layer 4 cells, in the case of cortex, of motor cortex, are also pyramidal cells, but they're stellate cells in the case of visual cortex. Here's a nice looking pyramidal cell, obviously, because there's not as far to go to the PL surface. The PL surface is the outside of the brain. The white matter is the projection on the inside of the brain. They are not as tall in terms of their apical dendrites. These are here in layer 6, corticothalamic cells. So it's described here as red L6CT corticothalamic, L6CC cortical. TT, what does that stand for? Okay, I don't. So we have these little circuits that we can build, and we can build one circuit on top of another circuit. So I'll try to see if I can identify something of the canonical circuit. So layer 4 receives inputs and now projects up to layer 3. That's the canonical bit. And then layer 3 should have outputs to layer 5. So layer 3 cell would be this cell. It projects to a PT cell here, projects to an IT cell here. So that would be the canonical circuit. And then these cells can project out of the local area. Classically, the intracephalic cells, whether of layer 2, 3, or 5, are going to go elsewhere in the telencephalon, and the pyramidal type cells will get out of the telencephalon, although they'll also project to the striatum, which is in the telencephalon. The corticothalamic cells will project out to thalamus. A pretty picture, but there's a lot of cells. Here we're showing just 25 cells. I have no idea how this picture was done, but it's cool. One of the studies that we did recently, and this was spearheaded by Cecilia, last name's not coming to me, Cecilia in Brazil, in collaboration with Salvador, with Antonio Roque and me. Romero, of course, Cecilia Romero. And she did this simulation in NetPyNE of a classical V1 model, Potjan-Diesmann model. It's been a very popular model, so we felt we should have it in NetPyNE. And we did a bunch of things with it to get a publication in Neural Computation, which should be coming out shortly. It just shows some of the beauty of the NetPyNE graphics, which we didn't fail to emphasize in the paper, were prettier than Potjan and Diesmann's graphics, but that was just rude. And Diesmann reviewed the paper, and he felt, yes, they are pretty. So we have raster plots of firing across different layers here. We have a picture, and we can, we could, if this wasn't in a journal, we could, of course, even get a nice movie of things flashing on and off. We have activity patterns up here, how much firing there is, and the different types of cells. And frankly, I can't read it, and I can't remember what it is. But we also have spectrograms. So here we have, over time, what frequency is active. And so we see activation here, a lot of coming and going of activity in the gamma range here of maybe 40 to 50 hertz. And here, similarly, but looking at particular projections and the dominant frequencies at those projections. Another project we're doing is dorsal horn of spinal cord. This was a project started by Kazutaka Seibuchi, who was a fellow visiting from Japan in the lab, and has been carried on by Laura. And again, I'm missing the last name. Laura can probably remind me of her last name. And she's at University of Toronto, and she's in this class. Medlock. Yes, thank you. Thank you, Laura. You remembered your last name. Good job. Sorry, Medlock. So and she's been doing a fantastic job, and Kazukata did too, and we're also working with Steve Prescott on this. Again, really mostly just trying to show the power of NetPyNE, put things together, get these raster plots, get the different frequencies, get the same kind of a connectivity matrix I showed earlier. One thing I failed to mention earlier is, of course, the connectivity matrix typically works with just that PIJ, probability of connectivity between different groups of cells, between different populations of cells. And it's just unclear to what extent you really need to understand what cell connects to what cell. Certainly, if you were trying to analyze a computer circuit, you would need to know actual wiring diagrams, not simply percentages. It's clear that there's degeneracy, right? Your brain and my brain are different, and yet for many practical things, such as seeing, such as speaking, we can do things the same, such as moving our finger. We do things very much the same way, despite having somewhat different circuits. So the actual individual to individual may not be important, and yet that is likely highly constrained during development in a way that we don't understand. And I somehow ended up with the same figure 3 times when I meant to get 3 different figures. So we'll just move on. Just to mention briefly that the cortex is not everything. So other areas of interest, an area that we've modeled, many people have modeled, the hippocampus. Again, this is where there's some disagreement between those who would prefer to work in a far more simplified vein and those who want to work with really highly detailed models, the hippocampus and entorhinal cortex. And this has become very popular because of place cells, because of the Nobel Prize given to O'Keefe and to the 2 Norwegians. They are very much spike coding oriented, and that's what they present in their papers. And so a lot of the modeling is done with very simplified point neurons at just scalar encodings. We've done a little bit of modeling of this, and we are actually doing a lot more now with the Blue Brain project. You're working with Michele Migliore in Italy. These are a very interesting set of projections. The classic Trisynaptic pathway goes from entorhinal cortex to dentate gyrus to CA3 to CA1. These are the Schaffer collaterals. And at the same time, you have this bypass projection, which is called the temporoammonic pathway, which projects more distally to CA3 and CA1 cells. And then this all closes a loop via subiculum back to entorhinal cortex. So it's a very interesting circuit. Again, the complexity is enormous. Just looking in this area, which I didn't even mark, which is the hilus of the dentate gyrus. David Amaral, who's a very well-known anatomist, looked at this area for his PhD thesis, and I think identified 25 different types of cells just in this tiny area, which is omitted here. So there you have it. The thalamus, I think, is super cool. And thalamus projects to different parts of cortex. Different nuclei do not project to each other, but project up to cortex. The exception to not projecting is the reticularis, which is kind of a pillowcase around each of these nuclei. The reticularis cell projects to the relay cell, gets projection from cortex, and there's this reciprocal connection which causes potential for oscillation, both a normal oscillation in sleep and an abnormal oscillation in epilepsy. I mentioned yesterday about the Trisynaptic pathway, so mysterious. The interneurons provide this end of a dendrite, which is all wrapped up in glia, and which then receives input from the retina. This is almost the first reception of input from the outside world, and then relays it as an inhibitory, presumably as an inhibitory input into the relay cell itself at the same time as it's receiving this excitatory input. So also you're getting this projection from the parabrachial region at the same time through a similar trisynaptic pathway. Mystery upon mystery. Very cool, very cool. Cerebellum, a favorite area because it has something to do with motor control, although other people, Jim Bauer in particular, will tell you it's really ought to be thought of as a sensory area. Abnormalities of cerebellum interfere with sensory processing as well. There's cases of, I think, dyslexia that involve the cerebellum. Apparently involve the cerebellum. Anyway, another very interesting, very mysterious circuitry. The famous Purkinje cells, which look so pretty. They're not so pretty in my picture because I don't draw that well, but they are enormous. They are the principal cells, comparable as principal cells to the layer 5 cells of cortex, right? But these are not excitatory cells. These are inhibitory cells. And so they are inhibiting these deep nuclei. That's where they project to. They can be activated enormously strongly by climbing fibers from the inferior olive. And they also can be activated by granule cell inputs, which come across like telegraph wires across these big dendrites. I think that's right. The granule cells are excitatory, I think. You also have mossy fibers from the spinal cord onto the granule cells. The Golgi cell, I don't remember very well. We have Ito cells, which we were just learning about from one of our collaborators. Basket cells, complexity. The basal ganglia we're starting to get interested in. We put in a grant submissions for this. Cortex famously projects to striatum at caudatum putamen, and anomalies in these areas lead to disorders, namely Parkinson's disease and Huntington's disease, as well as some dystonias. We have the direct pathway, which is, let's see if I can find it, cortex to striatum to globus pallidus pars interna, and then to thalamus. That's the direct pathway. The indirect pathway comes into striatum, goes to the pars externa of the globus pallidus, projects then to the subthalamic nucleus, then to pars interna, then to thalamus. Most of this circuitry is inhibitory, except subthalamic nucleus projections are excitatory. These little circles are a classic little code for inhibitory lines for excitatory. A lot of other things interest us, interest you, interest everyone. Retina has been modeled quite a bit. It remains a mystery. It's got it's got the dark current. It's got a current that gets turned off by light when it hits photoreceptors. It's got some non-spiking cells, amacrine cells, horizontal cells. One or both of those is non-spiking. It's got a complex layering. It's doing a lot of calculating. And, you know, people want to just understand, how does vision work in the cortex? Well, we probably need to understand the lateral geniculate nucleus and the retina as well if we want to know something about how vision works. Ventral horn of the spinal cord, I know Joe Graham's particularly interested in that. He's looked a lot at the cells there. Enteric nervous system is an entirely independent system. You can cut it off from the rest of the body and it will still function beautifully. It's basically like a little worm within you that's alive, just like in those parasite movies, horror movies. The intrinsic cardiac nervous system, something we just got started with in collaboration with Raj Vadakapali down at Jefferson University, and Jim Schwaber, and two of the people here, namely Dr. Gupta and Dr. Feldman, are involved with that. The claustrum we've been doing with one of the tutors, Erica, as well as with Jessica, who I just mentioned is Dr. Feldman. She's working on that. The superior Oliveira nucleus is a quite interesting part. A lot of the deep nuclei of the auditory system are interesting. The amygdala is interesting. You also have to consider as you go across organisms. Of course, as long as you stay in mammals, at least you can generally identify the same nuclei. When you get to the crab, you get a stomatogastric ganglion, which is not something you would identify explicitly in a mammal. When you get to fruit flies, I think we saw somebody's interested in fruit flies. Any of the invertebrates, obviously very different than mammals. Birds and reptiles are also very different than mammals, and you have to think about their circuitry differently. But even across mammals, if you want to study the auditory system and you decide you're going to study it in the bat, it's going to look very different because they do very special things with their auditory system, of course. So, yeah, I think I'll stop there. Take any questions. Or not. Okay. And now we will segue immediately or after 10 minutes into a tutorial, Salvador?
Yeah, I think we can start at 5.
Okay. Thank you all.
Thank you so much, Bill.
Thank you, Bill.
Thanks, Bill.
Thank you.